So today we're going to talk about the scientific method, which is a procedure used by scientists since the 17th century to discover natural truths through structured patterns of observation, measurement, experimentation, in order to formulate, test, and modify hypotheses. So, what does that mean? Well, that's the whole point of today. An observation and questioning is the first step of any scientific experiment. And you do this all the time, or you should. An observation is a remark, statement, or comment based on something someone has seen, heard, or noticed. The process of observing something or someone carefully in order to gain information. Now, you do this all the time. You see a bird fly or bat fly, like, oh, what's that? Why does it do that? That's observation and questioning all in one. If anything ever makes you go, ooh, what's that? Or, ooh, why does that do that? Or, then you're observing and questioning. Perfect. And we all do this naturally. It's part of the human nature to observe and question. Now, you can use your five senses, your sense of sight, sound, smell, taste. All of these and touch are used when you observe and help you to formulate questions. And that's highly encouraged. Keep doing that. Always do that. Even when you're 80, do that. So once you have a question, you've picked a question, you're like, I want to know about this. Then you need to propose a hypothesis. And a hypothesis is simply an educated, informed guess as to why something's happened. And your guess is not just based on nothing. It's based on observations. It's based on prior knowledge. And it will also become based on you doing what's called a literature search. You go and see what's already known about something or some process. And then you say, oh, well, we know this. We don't know this. And then you say, I want to see about this because no one's figured this out. And then you have your hypothesis. Usually a hypothesis is phrased as an if-then statement. So you say, if such and such happens, then this should be the result because we know blah, 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 blah. And you'll fill in the blanks. For example, if I give children sugar, then they will become hyper because sugar is a form of energy. So that is one hypothesis, and we're going to work with this throughout this video. Here, the sugar is your independent variable. That is the one thing you are choosing to control and test. This is something that you control and you choose. It's very important. If it helps, say, I chose the independent variable. I control the independent variable because independent starts with an I. Helps me, hopefully it'll help you. Next, we have to measure our dependent variable. This is the thing that you can measure and observe, but you can't control it. So in the case of the kids and the sugar, you would measure their level of being hyper. Make sense? All right. And as we know, kids kind of act like Tasmanian devils, or at least some of them do. I did. Maybe you're a good kid. So next we get to our procedure. How are we going to test this hypothesis in a way that is scientifically sound and accepted by other scientists? So what we do is we create the procedure, the step-by-step instructions for carrying out your experiment. So your procedure must be clear and repeatable by others. It has to be proven wrong, so it has to be something that we can measure and gauge. You can only use one independent variable, one thing that you're testing at a time, and this just helps to make sure that what we're testing is the cause or is the root cause and a cause and effect situation. So example, we might say we're going to look at the effects of sugar. We're going to give some kids 5 grams of sugar, some get 10, some get 15, and some get 20. And that's going to show us the effect of different degrees of sugaring these kids up. Now, some kids, actually all kids, come with a certain level of hyperactivity anyway, whether you gave them sugar or not, right? So we have to have some kids that get no sugar because we need to know what level of hyperactivity is just due to natural being a kid and what amount is because we gave them sugar. And that's going to be your control. In this case, it's a negative control because the kids in that control group are We're withholding sugar from them. They don't get it. And we know how they should act. Still crazy, but not as crazy. So everything else that you measure, or that you use, not measure, everything else that you use in the experiment, the size cup that you put the sugar in, the amount of water or flavoring you put into that cup with sugar or no sugar has to be the same. Everything else that you do in this experiment setup has to be the same. Same cup size, same amount of water, same time of day, everything. And those are considered your constants. And that's to make sure that you're only testing one thing at a time, that one independent variable. So then we give the kids the sugar, we let them be hyper, we measure their hyperactivity, however you decide to measure that, and we have our data. What do we do with this data? Well, data, first of all, is any information that you gather from your experiment. Now, there's two types of data. There's qualitative data, which is an approximation or qualities of something. It's not a number, it's anything but a number. If you're looking at coffee would be, qualitative data would be smell, the appearance, the heat, the color, the taste. Those are things that you can't put into numbers, but you can put into qualities or characteristics. Now, the other kind of data that we want to use in this class and other classes is quantitative data, data based on numbers. So same cup of coffee, if it was quantitative data with an N, you would look at how much coffee is there. You've got 12 ounces. The serving temperature is 150 degrees, another number. Your serving cup is 7 inches in height, another number, and it costs $4.95. So it's all about numbers, because we can take numbers and do what's called statistical analysis. And that's really important, which I'll tell you how to handle statistics in another video. But in general, statistical analysis is where you take your number data that's gathered, you sort it, you analyze it, and you put it into a graph. Because if you ever have seen data, a big spreadsheet full of numbers, it'll make your head blow up. And you just want to go, no, I don't, just go away. Nope, I'm tuning out. But instead, we can make it look fun and easier to read by putting it to a graph. A graph is simply numbers put into a picture, because as the saying goes, a picture is worth 1,000 words, and it's true. So on the graph, you can see, or on the PowerPoint, you see the straight up data of just numbers versus the graph. Same data, what would you rather look at? I'd rather look at the graph. And we're going to be making and looking at graphs, and you'll see how much nicer it is to interpret and understand what it means once we have that stuff graphed. So you have your data, you have your graph, you have your statistical analysis, and now we have to do what's called making an inference. An inference is simply an interpretation of what's been observed. So here's your information, what does it mean? What's the meaning of it? You got three options. You've got your hypothesis, right? And three things can happen in general. Your results can support your hypothesis, in which case you say, great, we're good. But you do still have to have it proven. So you have to do what's called peer review. So other people have to take your experiment that proved your hypothesis. They have to be able to run it, get the same results that also support the hypothesis. And once enough people do that peer reviewing of running the same experiment, they say, okay, yeah, this is legit. You're good. You did everything right. We're going to accept it. It may become a theory. It may become a law. But it's generally accepted by the public. Now, the second thing that can happen is that your results do not support your hypothesis. In that case, you reject your hypothesis. Okay, that's not really how it is. Still good information, still important to the scientific body of knowledge. But you might want to go back and reformulate a new hypothesis based on your results, and then set up a new experiment, new hypothesis, test a whole new situation. Good science leads to more questions than answers. Keep that in mind, because that's true and that's not always conveyed to students and the public. Now, your third option is that your results might partially support the hypothesis. Now, when that happens, you will probably revise your hypothesis, set up a new experiment, and run it again to see if you can get a more clear result. Now, be careful, because you don't want to bias your experiment by getting the end goal. Your end goal is not to be right, it's to find out the truth. And there's often a big difference between what's right, or being right for the sake of being right, and finding out what is true. So be careful. You always want to seek truth over being right. And in our experiment with the sugar in the kids, if your results support your hypothesis, you might say, we have found that the more sugar a child is given, the more hyper he or she acts. And therefore, sugar intake is a good predictor of hyperactivity in children. Reasonable if your data backs it up. So then you have to write a conclusion. You've got your hypothesis, you've got evidence for or against it, and now you have to summarize what you've done and what you found, because people don't like to read. I know it's a shock, people, but they don't like to read. So you gotta make it interesting, you gotta make it fun, you gotta make it to the point, because we got stuff to do. Ain't nobody got time to read 3 papers when you can say it in 5 sentences, right? I know you agree. All right, so a conclusion is simply a statement that summarizes an experiment and uses evidence to back up the summary. We've been trying to teach you guys how to do this for years. I think you can do it. So again, our sugar experiment with the kids. What we might say is, when given different quantities of sugar, 0 grams, 5 grams, 10 grams, 15 grams, and 20 grams respectively, and allowed to play, children ages 5 through 7 showed a positive correlation, positive link, of hyperactive behavior. That is, children given higher doses of sugar responded with significantly higher levels of hyperactivity than those given the control dose of sugar of 0. Now, we do have to talk later about what significantly means, which I'm not covering right now, but that'll be the next video. But as a hint, it's based on statistics, which you're gonna love. It's not as terrible as people make it out to be. So if you look at the graph in our made-up experiment, I feel the need to talk about the control a little bit more. So if you look at our made-up data, you have hyperactivity on the y-axis, which goes up and down. It goes from 0 to 100. On the x-axis, the horizontal axis, you have your control, your 5 grams, 10, 15, and 20. The bars indicate how hyper the kids are. So if you look at the control, you get 15%. Now, we can't attribute the control to any of the 5, 10, 15, or 20 gram hyperactivity, because that's natural. So you have to sort of delete or subtract that. And we've done that using this horizontal black bar that stops right above the top of the control bar and goes straight across. So everything above that black line is or can be attributed to the sugar given to the kids, but nothing below. And so that's the stuff that we can actually use as evidence of hyperactivity due to sugar. Hopefully that makes sense. If not, ask me questions tomorrow and we can go over it. Now, your homework is to look at the practice slide, which shows a cartoon of a child getting a shot in the arm. Observe the cartoon, propose 2 hypotheses as to why the child might be crying. Think back to when you were a kid and what frightened you and how things felt. Hopefully I didn't give too much away there. Then propose an experiment for one of the 2 hypotheses that you came up with. Use the PowerPoint here that we've gone over, and as a hint, there is no right answer, but there is a right procedure for your experiment. Try it, let's see what you come up with, and I'll see you tomorrow. Have a good night.